Assalamualaikum and good afternoon, everyone. So today we are going to continue our topic for this week, which is circulatory system. So let me share my slide first. Okay. I hope all of you can see my slide. So sorry for the date. Okay. So let me start our class. So on the hold on. So on the last lecture, we already learned about the function of the circulatory system. Okay, which is we also have the different types of circulation. That we have the open and closed. Okay, we also study about the circulatory systems in arthropods, insects, mollusks, annelids, and also chordates. Okay, and also include of the vertebrate. So today we are going to continue about the vertebrate hearts. Okay, so the vertebrate hearts it have complex walls with four main parts, which is the first one is the pericardium. Okay, we also have the epicardium, myocardium, and endocardium. All right. So as you can see from this figure, okay. So for the pericardium, so a pericardium is a sac uh, that surround the heart. Okay. So it has two layers, which is the parietal, the outer layer, and also the visceral, which is the inner pericardiums. And these parts, the pericardium parts of the heart, is filled with a lubricating fluid. Okay, so this is the pericardium layer. Okay, which is the content of the parietal pericardium, which is the outer part, and also we also have the pericardiums, which is the inner part of the pericardiums layer, right? So the second parts of the vertebrate hearts, we have the epicardium. So the epicardium is actually the outer layer of the heart that made of connective tissue. Okay, it continues with visceral pericardiums and contains of nerve that regulate the heart, and also the epicardium it contains of coronary arteries. Okay, so this is the epicardiums. Okay, it is actually the continuous with the visceral pericardiums, which is the inner part of the pericardiums. All right, in here. So the third parts of the vertebrate hearts is the myocardium. So the myocardium is the middle heart, the middle layer of the heart muscle. Okay, so as you can see from here, so this is the myocardiums, and the last parts of the vertebrate hearts is the endocardium. So we're talking about endo. Okay, so it is actually the innermost layer of the connective tissue that covered by the epithelial cells. Okay, we also call as endotheliums. Okay, so this is the endocardiums. Okay, so this is the endothelium's connective tissue. Okay, this tissue is covered the endocardium. All right, so this is the main parts, the four main parts of the vertebrate heart. So we go through the middle parts of the vertebrate heart, which is the myocardium. So myocardium is the ventricular muscles can be composed of two different types of myocardiums. Okay, so myocardiums composed of compact and also spongy. So compact is tightly packed cells arranged in a regular pattern. So this is the compact myocardium. Okay, so this is the pericardium. All right. So the pericardium also can be spongy, which is a meshwork of loosely connected cells. So this is how the spongy myocardium looks like. Okay, it's a meshwork of loosely connected cells. And the myocardiums, the relative proportions of the myocardiums are vary among species. Okay, for example, in mammals, mostly they have a compact of myocardium, and for example, in fish and also amphibians, they are mostly have the spongy proportion of myocardiums and arranged into trabeculae that extend into the heart chambers. Okay, so the proportions of the myocardiums are different. Okay, they are vary among the species. So we move to the cardiac muscles oxygen supply. So this is a working muscle that requires ATP. Okay, then the ATP requirement proportions to cardiac power output. So ATP is the means. Okay, it's a requirement. Okay, to the as a cardiac power output. Okay, so we have a spongy here, which is the spongy is a venous blood supply, and simplest but intricate designs, and the last organ supplied with oxygen. Okay, while for the compact, it is a coronary blood supply. It have a compact design and first organ supplied with the oxygen. Okay, so so this is the phylogeny and ontogeny. Okay, they are very depends on the species. For example, the hagfishes and lampreys, they have a spongy of myocardium or ventricular, okay, ventricle of the heart. Okay, so for the sharks and rays, they have a spongy plus variable compact. Antelopes, they have more spongy, but some have variable compacts. For amphibians and reptiles, spongy, and some of them they have a compact. Neonatal birds and mammals, most of them are spongy, and adult birds and mammals, 99% of them are they have a compact of the myocardium. All right. So this is the cardiac anatomy. So when we're talking about the cardiac, is actually related with the heart. Okay, the cardiac muscles, and they have a different number of chambers. Okay, so as you can see from here, for example, here for the fish heart. So for the fish heart chambers, they arrange in a series. Okay, they arrange in a series here. For amphibians and reptiles, they have three chambers, okay, of hearts. Okay, one, two, three here, ventricles, and also they have two atriums. And for the birds and mammals, okay, they have four heart chambers. Okay, they have two atriums, and also they have two ventricles. Okay. Um, so differences between the heart. Okay, for example, just now we are talking about the fish heart. They have one atrium and one ventricle, so it called as a single circulations. Okay, they only have two chambered heart, which is one ventricle and one atriums. And for the reptiles and amphibian hearts, they have two atria and one ventricles. Okay, so both oxygenated and deoxygenated blood are mixed. So it called as a double circulations here. So they have two, this reptile and amphibian, they have three chambered hearts, which is two atria and one ventricles. Well, and the oxygenated and oxygenated blood are mixed in the ventricles. Okay. While for mammalian hearts, they have two atria and two ventricles. So the oxygenated and deoxygenated blood are separate. Okay, so we also call as a double circulation. So as you can see here, so they have a four-chambered heart. Okay, so because of they have two ventricles, so the oxygenated and deoxygenated blood they separate between the two types of blood. Okay. So this is the mammalian cardiac cycle. So this is the steps how the cardiac cycles. Okay, so you can read it after this. Okay, they already state that this is the step one. Okay, step two, step three, step four, and step five from the ventricle diastole to the again to the ventricular diastole. Okay, so this is the cycle. Okay, you can read it after this. So we move to the heart pressures. Okay, so the two ventricles contract simultaneously, and but the left ventricle contracts more forcefully and develop higher pressure. So you can see from this graph. Okay, so this is the left side of heart. So the left ventricles contract more forcefully and more develop higher pressure. Okay, as you can see from here. So this is the left ventricle compared to the right ventricles. Okay, and the resistance in the pulmonary circuit is low due to high capillary density in parallel. Okay, so you can see from here. So they have low the resistance is low here, and they also have less pressure is needed to pump blood through this circuit in the right side of heart. Okay, so the low pressure also protects the delicate blood vessels of the lungs. So the pressure, this is the pressure changes in the hearts and arteries of mammals such as humans. Okay, so the left side here of the heart which supply the systemic circuits. Okay, so it develops substantially greater pressure than the right, because of the right is supply the pulmonary circuit. Okay, so they have a low due to the high capillary density. Okay, so this is the initiation of cardiac contraction. Okay, permulaan untuk cardiac contraction. So we have three here, which is neurogenic pacemaker. So the neurogenic pacemaker is actually the rhythm is generated in neurons. Okay, for example, in some invertebrates. Okay, the heart, the cardiac contractions are generated in neurons. While we also have the second one is the myogenic pacemaker. So, for example, in vertebrates and also some invertebrates, okay, include of humans. So the myogenic pacemaker is the rhythm that generated in myocytes. Okay, so this is a pacemaker. Okay, and the we also have the artificial pacemaker, which is the rhythm are generated by device. Okay, so this is the initiation of cardiac contraction in our heart. So how to control the contractions? Okay, so vertebrate heart are myogenic. Okay, just now you're talking about the myogenic pacemaker. Okay, the last slide. So the myogenic pacemaker is the rhythm that generated in myocytes. Okay, so vertebrate heart are myogenic. Their cardiomyocytes produce spontaneous rhythmic depolarizations in here. So the cardiomyocytes are electrically coupled with gap junctions to ensure coordinated contractions. Okay, so pacemaker. Okay, we're talking about the myogenic pacemaker. So pacemaker is actually the cells with the fastest intrinsic rhythm. Okay, the naturally rhythm. Okay, so pacemaker is the cells with faster intrinsic rhythm. Okay, so the initiate the heartbeat. Okay, fish is located in the sinus venuses, and other vertebrates are located in the sinoatrial or SA node in the right atriums. Okay, so this is where the location of the initial of the heartbeat. So the we go to the myogenic contraction. So all cardiomyocytes can contract without an external stimulus, and resting member potential is unstable. So we call as the pacemaker potentials. Okay, here. So this is slightly unstable here, and specialized cells. Okay, we call as a pacemaker. Okay, so pacemaker is the cells that fastest intrinsic rhythm. All right. So the specialized cells, the pacemaker, set the intrinsic heart rate and the relative timing and speed of the opening of a specific ion channels, which is we have the sodium ion channel. We also have the calcium ion channels, and also we also have the potassium ion channels. Okay, that relate with the time and speed of the heart rate. Okay, so we go to the first one, which is increasing the heart rate. So the hormone that involved in this process, the increasing of the heart rate, is the norepinephrine. So the norepinephrine is released from sympathetic neurons, and also we have epinephrine hormone. It is released from the adrenal medulla. So more of the sodiums and also calcium channels will open, and it will increase the rate of depolarizations, and also it will increase the rate of action potentials. Okay, so this is the action potentials. And how about decreasing the heart rate? So the hormone that involved in decreasing the heart rate is the acetylcholine. So this acetylcholine hormone is released from parasympathetic neurons. Okay, so in here, the specific ion channels that involve is the potassiums. Okay, so more potassium ion channels will open, and the pacemaker cells will hyperpolarize. Okay, compared to the increasing of the heart rate, the rate of depolarization will increase, but in decreasing the heart rate, the pacemaker cells will hypopolarize, and time for the depolarization will take longer. Then it will decrease the heart rate. Okay, so we see the steps. Okay, the effect of the first one, which is increasing the heart rate, the effect of the norepinephrine on heart rate. Okay, so it detected by the cardiovascular control center, the medulla. So from this medulla, it will release the norepinephrine from sympathetic neurons. Okay, and also releasing of the epinephrine from the adrenal medulla here. Okay, so either the norepinephrine or epinephrines. Okay, so from the extracellular, it will bound with the receptor, beta 1 receptor here. It will bind with this receptor, and after the binding, it will cause the the increasing of the sodiums and calciums uh, channel to opens. Okay, and it will increase the influx of these two ions. Okay, the sodiums and also the calciums. Then it will increase the rate of depolarizations. Okay, then after the increase of the rate of depolarization, then it will increase the heart rate. Okay, so the effect of acetylcholine hormone on the heart rate, okay, it will decrease the heart rate. Okay, so it detected by the cardiovascular control center, and it will release the acetylcholine hormone by the parasympathetic neuron. Okay, so from here, it will bound to the receptor. Okay, the type C calcium channel. Okay, the receptor here. Sorry. The acetylcholine will bind with the receptor, which is muscarinic receptor here. Okay, so it will cause the potassiums. Okay, the potassiums channel will open. Okay, and will decrease the calcium's influx. Okay, it will decrease it will decrease the uh, calcium ions, okay, influx daripada masuk. Okay, so this, okay, so it will take the time of depolarization will take longer. Then after that, it will decrease the heart rate. Okay, from this hormone of acetylcholine. Okay, So this is the effect of acetylcholine on the heart rate. Okay, so we move to the regulation of pressure and flow. The circulatory systems must regulate the distributions of flow to the tissues. All right. So highly aerobically active tissue have a greater demand for oxygen than do the less active tissue. This require it will demand and it will increase the blood flow. Okay, so the pressure, resistance, and flow are varied throughout the circulatory systems and in response to changing conditions. Okay, it depends on the demand. So arterioles. Okay, so here the arterioles play the primary role in distributions of flow within the circulatory systems because these arterioles can be vasoconstrict and vasodilate. Okay, in the previous lecture we are already talking about the vasoconstrict and vasodilate. And the arterioles are arranged in parallels, okay, and the blood can be redistributed as needed. Okay, so for example in here, okay, during exercise, the arterioles of the gut and the kidneys will vasoconstrict, okay, whereas the arterioles of aerobically active skeletal muscles will vasodilate, okay. Thus, it will decrease the flow to the internal organs here, for example, gut and kidney, and it will increase the flow to the active skeletal muscles, okay. So it means that where is the higher demand, they will have a higher blood flow. Okay, for example, during the exercise, the skeletal muscles demand more blood flow compared to the internal organs. Okay, so this is metabolic activity of tissue. So the table shows the factor that influencing the vasoconstrictions and vasodilations. Okay, so this is the substance for vasoconstriction. So this is for vasodilations. Okay, we have exposure. Okay, this is the source and the type. Okay, you can look through this table after this. So okay, so this is the response of arteriolar smooth muscle to an increase in metabolic rate. Okay, so we have a process of negative feedback here. So if there is a higher demand, okay, in the tissue metabolic rate, so it will decrease the oxygen and increase of the carbon dioxide and the waste. So it will send the information to the arteriolar smooth muscles. So the arteriolar smooth muscle will vasodilate, okay, and they will have decreases in resistance and they will have the higher blood flow. Okay, higher demand, they will have higher blood flow. Okay, so so it will increase the delivery of oxygen, and also it will increase the removal of carbon dioxide and also waste. Okay, so then the level of oxygen in the tissue will increase, and also it will decrease the level of carbon dioxide and also waste in the tissue. Okay, so then it will go back to the normal one. So it will stop the flow from increasing beyond what is needed. Okay, so if less demand, so it will have lesser blood flow. If it will have higher demand, so it will have higher blood flow. All right. So we are talking about blood pressure. Okay. So how to measure the blood pressure? So blood pressure measurement is the measurement of force applied to artery walls. Okay. So this is how we measure the blood pressure. So the pressure fluctuations are largest at the heart, okay, and the degree of fluctuation decrease through the circulations. Okay, they are decreased through the circulation. So we have the systolic pressure. Okay, in the last previous lecture we're talking about the systolic pressure, diastolic pressure. Okay, so actually the degree of fluctuation is decreased through the circulation. And also, as you can see from this graph also, they have the highest of fluctuation in the heart, okay, in the left ventricles. Okay, so this is arteries, the pressure reservoir. Okay, so the aorta acts as a pressure reservoir and dampen the pressure oscillations caused by the pumping actions of the heart. Okay, if the pumping action, the ventricular contractions, so the blood flow will they will have a rapid blood flow and it will, but when the ventricular are relaxed, okay, the ventricular relaxations, so the blood flow kisses and reduce the aortic pressure here. Okay, so this is the in the arteries, okay, the pressure of reservoir in the arteries. So moving blood back to the heart, okay. So the just now we're talking about the arteries. So here in the veins, so blood in veins is under low pressure. Okay, so we have two major pumps assist in moving back blood back to the heart. The first one we have skeletal muscles. Okay, as you can see from here, so they have a valve here. So this is when the skeletal muscles are contracted. Okay, so the valve here will close and the valve it will open the valve to the heart. So the blood will flow back to the heart. Okay, but when the skeletal muscles are relaxed, so the valve here will close. Okay, so the valve here will open, so it will have the continuous of pumping here, the skeletal muscle here. Okay, so we have the second pumps that assist in moving blood back to the heart, which is the second one is the respiratory pumps. Okay, so we have the inhalations and exhalation. Okay, so when we inhale, the pressure in the thoracic cage will drops and draw blood into the veins. Okay, and when we exhale, the pressure increases in the thoracic cage and pushes the blood towards the heart. So the blood does not move backwards because of the valve. Okay, so this is the two major pumps that assist in moving the blood back to the heart. So this is the lymphatic system. In the previous lecture, I mentioned a bit about the lymphatic systems. So the lymphatic system is plays the important role in fluid distributions. All right. So so we have different type of fluids, okay, that need to be distributed throughout the body. So for example, we have blood, we also have hemolymph, okay. I'm talking in the previous lecture. So this lymphatic system also important is an important part of the immune systems, okay. So some fluid leaving the capillaries and enter the lymphatic systems. So this fluid we call as the lymph. It flows through the lymph nodes. Okay, so this is the lymph nodes. It flows through the lymph nodes and lymphatic ducts. Okay, and the lymphatic ducts and returning the venous part to the circulatory systems. Okay, near the right arteriums, and the lymphatic ducts contain the valve. The valve there. Okay, contain the valve here that ensure the unidirectional flow. So the lymph does not flow back to the capillaries. Okay, so this is the relationship between the mammalian circulatory system. So this is the circulatory systems and the lymphatic systems. Okay, so the effects on gravity on the blood pressure. Okay, so the measured blood pressure when standing includes a hydrostatic uh, gravity or hydrostatic component. Okay, when we measured our BP during during standing, so it will include the hypostatic component, which is the hydrostatic pressure. Okay, so but what happened when we change the body positions? Okay, when prone. So when measured the blood pressure when prone, you can see here. So the heart rate is 13 from 6 kp APA, while during standing it will less. It will have less. So what happened when we change our position? What do you think the position of the body may affect so do you think that the body positions may affect the BP readings? Okay, so what do you think? What is the best positions to have to measure the accurate BP? Either standing or sitting or lying. Okay, so you can trying to figure out about this, the effect of gravity on our blood pressure. So just now is the effect of gravity to our blood pressure. Then and this is the effect of gravity on the blood circulations. Okay, how does the gravity affect the blood circulations? So in here we have two example here. So we have the giraffe and also a human. So very tall animals, for example here giraffe, they must be able to pump the blood up to the head. Okay, so they could also have difficulty with blood pooling in the feet and peripheral edema. So it is different with the human. Okay, because of they don't need really hard pressure to supply to pump the blood throughout the body. Okay, it compare to the tall animals. Okay, so so you can see here. Okay, so this is the giraffe just now we are talking about. So when the giraffe stand, Allah Akbar, uh, so when the giraffe are standing here, so they will have the blood vessel will have the vasoconstrictions of vessels in the lower body. Okay, then the when the giraffe are changing the positions, so the aortic pressure will decrease. So the vasodilation of vessels in the lower body. So it means in the previous lecture we are talking about the vasoconstrictions. Okay, right? So the vasoconstrictions in here it refers to the narrowing of arteries and blood vessels. Okay, so during the vasoconstriction in here, so the heart need to pump harder to the body. Okay, through the constrictions veins and arteries. Okay, so this can lead to high blood pressure. Okay, during the vasoconstrictions, while during the vasodilations, so it is the widening of the blood vessels. Okay, so it results from relaxations of smooth muscle cells within the vessel walls. Okay, in particular in the large veins and also large arteries and also the smaller arterioles, because they don't need a higher pressure to distribute the fluid throughout the body. All right. So we move to the blood. Okay, the last subtopic for the circulatory system, which is the blood. So the composition of the blood, primary, it contains of primary water that containing of ions and organic solute. It also contains of blood cells and also protein. Okay, in our blood. So the blood protein in vertebrate in the invertebrates are primarily respiratory pigments, and normally they use to transport or to store the oxygen. Okay, the protein in the invertebrates, while for the vertebrates it carries a protein such as albumin and globulins. Okay, and also they also contain of proteins that involve in blood clotting. Okay, in vertebrates. So we move to the blood cells. Okay, so just now the component of blood, the protein, so this is the blood cells, or we also call as the hemocytes. So the blood cells are functions in oxygen transport or storage again, and also functions in nutrients transport or storage, or and also phagocytosis, immune defense, and blood clotting. Okay, so this is the functions of blood cells. So we have different types of blood cells. Okay, for example, in insects, okay, in Drosophila, they have the plasmatocytes, they have the lamellocytes, they also have the crystal cells. And while in the vertebrates, for example, in fish, frogs, and mammals, they have the erythrocytes, okay, or we also call as the red blood cells, okay. Listen. So they also contain of lymphocyte, monocytes or macrophage, okay, and also they also contain of granulocyte and also thrombocyte. So all of these are called as the leukocytes, or we call as the white blood cells, okay. Okay. So we move to the erythrocyte, which is red blood cells. So the red blood cells are the most abundant cells in the blood of vertebrates. Okay, so it contains high concentrations of respiratory pigments such as hemoglobin, and the major functions of the erythrocyte is the storage and transport of the oxygen. Okay, and the erythrocytes have evolved independently several times. Okay, so the morphology of the erythrocytes, they have a round and oval or oval in shape. Okay, and also for the mammalian erythrocytes, they are biconcave discs, and these shapes increase the surface of area and facilitating in oxygen transfer. Okay, so this is the morphology of the erythrocytes in mammal and also in the other organisms. So just now we're talking about the red blood cells. So this is the leukocytes, which is white blood cells. So, sorry, okay. So leukocytes is the white blood cells, and it function in the immune response. So, and all of the leukocytes are nucleated, which is having a nucleus. Okay, and the leukocytes are found both in the blood and also in the interstitial fluid, and some are able to move across capillary walls, and they have five major types of leukocytes. So the first one, we have neutrophils, we have eosinophils, we have basophils, we have monocytes, and also macrophage. So this is one, and the last one we have lymphocyte. So neutrophils is uses phagocytosis to engulf the damaged cells. Okay, and also microorganisms and other foreign particles. So this is very importance of leukocytes, which is it will use the phagocytosis to engulf the damaged cells, the microorganisms, and other foreign particles. And for the eosinophils, okay, the functions is to deliver the cytotoxic chemicals and enzyme that kills parasites. So these eosinophils also are involved in allergic reactions. Okay. So the second types of the third types of leukocytes is basophils. So the function in immunity is they leave the circulatory systems and accumulates at sites of infections. Okay, and it will release the cytotoxic chemicals that kill the foreign particles, and these basophils are involved in inflammations. Okay, then the fourth types of leukocytes is the monocytes. Okay, so monocytes is a monocyte matured into phagocytic macrophage here that engulf and destroy the foreign particles and dead or dying cells. Okay, so this is the functions in immunity of monocytes. Okay, so the last one is lymphocyte. Okay, so the functioning in okay, so the lymphocyte, they have multiple types of lymphocytes, which is for example, the first one, the B cells. The B cell is function to secrete the antibodies, and the second types of lymphocytes is the helper T. They help to activate other lymphocytes, and also we have killer T cells or cytotoxic T cells. So it function to secrete the cytotoxic agents that kills invaders or dying cells. So the leukocytes are really important in the immune response. Okay, so the thrombocytes. sorry, the thrombocyte. So the thrombocytes play a key role in blood clotting. So the thrombocytes are spindle-shaped cells in non-mammalians and also classified as leukocytes, okay, in the non-mammalians. And also the thrombocytes are nucleated cells fragments in mammals. Okay, so in mammals we call platelets. So they have three steps in blood clotting. The first it will vasoconstrictions, then platelet plug will form. Okay, platelet plug formations, then the clot formations through coagulation cascade. Okay, so this is the blood cell formation. So we call as the hematopoiesis. Okay, the process of blood cell formation we call as the hematopoiesis. So it location of the stem cells. In adult mammals, only in the bone marrow, in the fishes, in the kidney, in amphibians, reptiles, and birds. Okay, the process are located in the spleen, uh, liver, kidney, and bone marrow, and the specific signaling factor are involved, okay, in this process, the process of blood cell formations. For example, we have erythropoietin, which is a hormone that released by a kidney in response to low blood oxygen. Okay, they involve the specific signals in the blood cell formations. Okay, so we already finish of the secretary systems from the last 2 days, which is in the Monday, and just now we are continue about the secretary systems. Okay, so I'll show you a video, 3 minutes video that give an overview of different types of secretary systems in different types of animals that we already learned throughout this week. Okay, so this is very, very catchy video. Okay, video kanak-kanak sikit. So let's see the video. Okay, that's all for this week lecture and for today lecture. So I'll see you next week. Okay, next week we will have tests. Okay, so let's be prepared. Okay, and if you guys have any uh, questions regarding throughout our semester, okay, starting from the week 1 until this week 5, just let me know. All right. So and okay, that's all. Thank you very much. Yeah, yeah, yeah. Okay, okay, okay. Wait, wait.